So good evening, everyone, and welcome to Flanders Vaccine's second COVID-19 webinar. Last week, we mainly focused on the preclinical and clinical research for the development of a preventive vaccine, but today we will also discuss the potential of some drugs for the treatment of COVID-19. For those who didn't join the webinar last week, my name is Fran van Heuverswin and I'm the project manager of Flanders Vaccine. And just as last week, I would like to start to thank all of you for joining us this evening. And I also would like to thank the speakers of this session, all Belgian scientists, medical doctors, and members of Flanders Vaccine. I really appreciate that they took some of their precious time to participate in this webinar. They are all working very hard in this very busy period, so thank all of you for joining us tonight. So far, there is no approved medicine for COVID-19, and there is no cure available. So scientists around the world are working very hard on potential treatments and vaccines for this new coronavirus disease. Some companies or research groups are working on antiviral drugs. This is also the case for the Flemish Institute for Biotechnology, the VIB in Ghent. In some of their projects, antiviral drugs, which are already used against other diseases, are tested to treat COVID-19 patients. Other scientists are working on the development of a vaccine. So in today's webinar, some antiviral drug projects will be highlighted. The potential of the use of an intranasal vaccine will be discussed, and we will also learn more about the role of adjuvants in the development of a COVID-19 vaccine. Unfortunately, Dr. Kai Dahlmeier had to cancel his participation, so this causes a little change in the agenda. So now, before I pass the word to Matthijs de Blok, the host of this meeting, and Vera de Kolvenaar, who will moderate the Q&A session, I will briefly tell you a little bit more about Flanders Vaccine. So Flanders Vaccine is a non-profit cluster organization. So we're trying to connect all the different stakeholders from academic, industrial, and public stakeholders who are active in the field of human and animal immunotherapeutics and vaccines. Next slide, please. So what is our mission? So what are we aiming at? We want to stimulate partnerships. We want to support innovation in the sector. We provide coordination across the relevant actors and sectors. We also want to enable a positive environment, especially for the conduct of clinical trials. And with all the different stakeholders, we want to build a strong ecosystem, and we want to position this ecosystem as an ecosystem of choice for other countries. Next slide, please. So how are we doing this? We do this by the sharing of knowledge and expertise. For example, this webinar, the organization of this webinar is a good example. We also participate in collaborative projects. We would like to improve the communication and raise awareness about benefits and risks of vaccination. We also have a strong collaboration with the Center of Excellence for Vaccines. This is a center of the Federal Agency for Medicine and Health Product in Belgium. Together with them and the 3 vaccine clinical centers, we also work on simplification and standardization of documents for the application for a clinical study. We also open for collaboration with other networks, and of course, we want a stronger branding in our region. So next slide, please. So if you want to collaborate with us, you can do this as a member. You can be a strategic partner, or you can sponsor one of our events, and we can maybe also work together on a specific project. So more information can be found on our website, or you can send an email to info@flandersvaccine.be. So then now I want to end this presentation to present the Flanders Vaccine team. So at the top, you can see our board of directors. These are also the founders of Flanders Vaccine. I also want to thank our strategic partners for their support, MSD, Janssen, GSK, Sanofi Pasteur, and Pfizer. And with the last slide, I want to show you all our members, of course. I also want to thank them for their continued interest in our organization. So here you can also see if your company or your organization is a member of Flanders Vaccine. So thank you.
So thank you, Fran, for this introduction. Before we announce the speakers, I want to share with you some of the best practices in this webinar. And within this webinar, all the participants, are in listening only mode. So you are able to hear us, but we can't hear you. If you have any questions, don't hesitate to ask them. We would love to have an interesting discussion afterwards. You can ask your questions by clicking on the Q&A button in the black menu bar on your Zoom interface. Just hover over your screen to see the menu. There you can write down your question. Please also write the name of the speakers before the question so we can direct them to the right person. We will cluster all the questions and ask them to the speakers after the presentation. We apologize in advance because it is possible we will not be able to answer all the questions due to lack of time. The webinar will also be recorded and will be shared with all the participants a few days later. So for the timeline, after this intro, we are happy to welcome 4 experts who will give a presentation of each 15 minutes. And after the presentations, we will have the Q&A sessions. So for the participants, again, don't hesitate to ask questions during the presentations. The more questions, the more interesting the discussion afterwards will be. After the Q&A, there will also be some closing remarks by Sven Arnouds, the chairman of Flanders Vaccine. So now, the interesting part, the experts. For this, I will leave the word to Veerle de Kovenaar.
Thank you, Matthijs. So the first speaker of today will be Wim Tiest. He's the head of infectious diseases at eTheRNA, and he will give a talk on the development of an intranasal vaccine to protect for COVID-19. Next, Professor Xavier Salens from the Center for Medical Biotechnology at the VIB and University of Ghent will share his insights on nanobodies as a potential antiviral drug against SARS-CoV-2. Then the third presentation of today will be given by Professor Bart Lambrecht, medical doctor at the University Hospital of Ghent and department director at VIB. But first of all, Professor Lambrecht, we would like to congratulate you on the FW Excellence Prize for Distinguished Career that you received yesterday. And thank you for tackling the repurposing of rheumatism drug for the treatment of COVID-19 patients during your talk today. The last speaker of today's webinar is Dr. Jamila Louahet, Vice President of Head of R&D Center Belgium at GSK. Dr. Louahet will share insights on the case of adjuvants and the acceleration of vaccines R&D. So the first speaker of today is Wim Tiest. So Wim, please start sharing your screen, and the floor is yours for the next 15 minutes.
But first, thanks of all. Thanks to the organizers for having me today in this very interesting symposium. I think last week was already a big success. We're going to try to do our best to make this one at least as good as last one. So, and I'm starting off, so that's fun. What I want to do today is share with you a story of a somewhat different type of vaccine. As you will see during the presentation, eTheRNA is not really doing what most of the other competitors in the vaccine field are doing in this COVID response. And so I'll try to talk you through that and explain a little bit more why we are doing what we're doing. So I guess most of you do know eTheRNA. We're a small Belgian vaccine biotech. We're built around messenger RNA technology. That's what we do. Most of our published data and also our active clinical research is in therapeutic vaccines in oncology. So that's really what most of the team, most of the research work is being oriented towards. So that means that as such, we do not really have the muscle for a massive, fast-paced effort like some of the competitors in the field are doing. And we didn't have a prophylactic platform that was ready to go. That was a sort of plug-and-play format to make a new vaccine against corona. But we were challenged in January by our investors and our management, and they told us, look, try to come up with a relevant solution despite all these constraints, because we really do believe that with our technology, we do have a strong fit to make a vaccine against corona. And so actually, we started to work on that. Now, we took a step back and we kind of forgot what we were doing, the technology, the existing platforms, and we asked ourselves, look, what would a good vaccine against COVID-19, what would it look like? But first of all, would need to be something that offers a long-term protection. Long-term protection, ideally also based on immune memory, not just on inducing antibodies that then wane over time. Now, why is that important? Because we are obviously talking about mass vaccination. We need to reach a maximum of people with this vaccine. So if we need to get into a repeated booster regimen where people need to come back regularly and be reboosted, that's going to make it even more difficult to reach coverage. So long-term protection seemed to us like a very nice first feature. We also decided that let's try to go for broad protection. And broad, I'm using the word actually in two senses. First of all, broad, because we want to involve all the different arms of the immune system, both T cells and B cells. And we're also thinking about using not just systemic immunity, but also mucosal immunity, because it's an airborne pathogen. And second, broad, because we do believe that it would be nice to build in a safety margin against either viral drift or viral variations. You can tell me, okay, we haven't observed any big variation in the current corona strain, but as you all know, it just takes one well-placed mutation or one well-placed crossover that is very efficient in replication, and it is happening. And I think the statistics today, the number of infectious cycles that are happening across the world are not really helping us to avoid this phenomenon. So I think we're better sure than sorry, and let's try to build in some margin into the vaccine. Third and fourth actually is the ease of administration. We are facing a vaccine that needs to be used across the world, that needs to go in all type of regions, that needs to reach a 60 to 70% coverage or even more. So ease of administration and the logistics behind it are going to be one of the key elements to succeed. And also we think that our effort and the effort that all the others are doing should contribute to solutions that can serve the next pandemic. We're not hoping that the next pandemic will be around next year, but I think the past learns us that it's probably the right time to make a well-founded effort and to make sure that whatever we do now can be reused when we need it the next time. So these were our starting points. Now you can say, okay, that's all very nice. How are you going to start about that? You're a small company, you're focused on oncology, but you didn't really start with a completely empty backpack. Actually, our platform, the mRNA platform that has an embedded adjuvant system inside it called Trimix, does induce very strong T cell response. And that's something that has been published both in ex vivo studies with dendritic cells, but also we have seen these responses appearing again in our own clinical studies. And on top of that, the safety of the product is excellent. So that as technology is a good start. Additionally, and that's something I would say of a luxury in the world of messenger RNA, eTheRNA has the in-house capabilities to design, to produce, and to scale GMP quality messenger RNA. We can do that in our Niel facility close to Antwerp. Don't ask us to produce 1 billion doses tomorrow. Obviously, we don't have the size and the facility for that, but we can certainly supply big clinical studies. We can certainly supply an entry to the market. On top of that, when we started the project, we very quickly reached out to a number of partners. And we were very lucky to start talking with people like Neil from Ghent, who have the tools to rationally divine the antigen needed for a CoV-2 vaccine. We also ran into a couple of very good preclinical CROs that can do all the animal work. We're working with immune experts who can do some in vitro validation. So we have surrounded ourselves with a number of partners, both inside and outside Belgium. On top of that, we met the enthusiasm of Pierre Van Damme and his Center of Vaccine, who are willing to help us and act as a principal investigator in this adventure, so we can build on his broad expertise in clinical and in pandemic clinical research. So certainly not a bad picture to start with. So actually, what is the scientific hypothesis that we are working on for this vaccine? We think the nose is the primary line of defense, and we try to focus our efforts on that part of the body for the vaccine and for the protection. Why are we saying that? Actually, there's a number of publications that have been coming out over the last years, not necessarily all with corona, some yes, but mostly in fields like influenza, that show that actually the nasal tissue resident memory T cells, and that's both CD8 and CD4 cells, can actually protect from lower respiratory tract infection. And we all know that the lower infection, that's the one that causes the disease in the patient. So the idea is if you can use those T cells to actually stop the virus at the point of entry in the nose, and you can avoid that it replicates wildly and that it spreads to the lungs, you do actually have a solution to stop the disease. It is not going to be a sterilizing vaccination. It's not going to be a neutralizing antibody that stops any virus from entering any cell, but it should stop the disease. And that's probably what's the most important, the disease, and hopefully also the transmission to other subjects. There's a number of publications, most focusing on CD8. We also found quite some very nice references that put a critical role for CD4 cells, also tissue resident memory CD4 cells. We know that the type of vaccine that we're going to put together is going to be very critical in stimulating this kind of response. We know we need a very strong Th1 and Th17 inducing vaccine. Fortunately, that's something that with messenger RNA and a strong adjuvant is perfectly feasible. And we also hope that inducing this memory can also help an antibody response against the virus later on or during the course itself. What is very interesting with this nasal and local mucosal immune responses is that it's very long-lived, where most of these cells need to be replenished from lymphoid stores during their cycle of life. From the publications, it seems that these cells, they are resident and they stay resident. So they can be around and protect for a very long period, which actually answers to the question of long-term protection. And there has been mechanisms based on this principle that have been shown with coronaviruses. So it's not just for influenza. There is certainly evidences that it can also work for corona. So that's the setting. Now, how did we define our candidate around those? Well, first of all, a broad immune protection. What we want to do is to focus on the best and the most conserved CD4 and CD8 epitopes. For that, we're not looking just at the S spike protein like most of the other vaccine developers are doing. We have actually looked at the whole viral genome and using tools, in silico tools like the ones offered by a partner like MyNeo, we have been looking for those epitopes, both CD4 and CD8, that first of all are likely to be best picked up by the immune system. But we also did the analysis in terms of protection, of cross-protection or potential cross-protection across the family of coronaviruses. So we didn't only look at the sequences for CoV-2, we also compared it with SARS-CoV, we compared it with MERS, we even looked at some of the common cold coronaviruses, and we made a mix of all these epitopes to be able to present a construct that has the most likelihood to be cross-protective, but also features all the best epitopes from the current corona strain. So that's one. The other is the intranasal delivery. We believe that going that route, it is a sort of topical administration. There will certainly be some debate with regulators on that definition, but the safety hurdle is definitely going to be lower than for an injectable. It will also induce the immune response exactly in the right spot. We do want the nose to act as a first barrier, so that's where we're inducing the responses. Ease of administration is obvious. In many cases, it could be self-administration, which could be a major benefit for a large vaccine campaign. And there is evidence and devices. There is FluMist on the market in the US, which uses a nasal atomizer and perfectly manages to immunize with that. And in fact, the use of RNA in the nose is also supported by published data, and it has been shown that it can induce strong T cell responses. Now, we want to position it as a platform technology. What does that mean? It means that potentially this could work for a number of viral pathogens or airborne pathogens, but definitely our initial focus is on corona. In terms of manufacturing, also to act as a platform, what we would like to achieve is that we can condition separately the excipient, the adjuvant, and the antigen. What does that mean? That means that we could have a much more flexible storage and stability program. We could stockpile actually excipient and adjuvants as a preparedness for a next wave or a next use, because the antigen would be the only part that needs to be changed over. In case the coronavirus changes and there's another threat, that is why this could serve as a future platform for pandemic preparedness. Now, what are we doing with that for the moment? This is in very short our development roadmap at eTheRNA today. Actually, in green, those are boxes that we are doing sort of independent of COVID. The blue boxes are really focusing on the corona antigen. So in terms of intranasal development, we are currently following two routes. In terms of excipient, we have a lipid-based and a polymer-based administration system that are both being tested, and we are trying to pick the best candidate for the next step. How are we doing that? We're doing that in a preclinical validation, and I'll show you one picture of the nasal targeting and the expression of the RNA. And in the next step, we're actually engaging, we're using flu as a model to check for immunity and also a challenge model to see if we can induce the right responses and if those can be protected. So that's really going on in parallel. In parallel of that, actually, we're doing the design of this CoV antigen, where we're picking CD4, CD8 epitopes and reassembling them in a concatenator, best conserved, coming from SMNE protein. And that construct we will validate in vitro. We will see whether technically we can make it, whether it's expressed, and whether actually it can be recognized in vitro, and those epitopes can actually be picked up by the immune system. That should be done by September. Then we should be ready to start GMP production of the RNA, and also to complete our preclinical tox package. Now, there the timelines are a little bit uncertain for us because it will depend a lot on the choice of excipient we make. Within the two families, there's one which has rarely been used in human before, and there's another one which has already been in phase 1 studies in another setting. So if that one turns out to be the best candidate, actually the second step, GMP in preclinical tox, could be relatively short. And then we anticipate to do two things in parallel. We will certainly do a human volunteer study to check for safety and for the immunity that we induce, specifically the nasal immunity. And in parallel, we would like to run a challenge model. We discussed at length challenge models over the last weeks, also in sessions of the last webinar. We're eagerly waiting the selection of the most relevant model, and that's certainly the one that we will also pick up for this parallel step of development. Now, I'm going to share one picture just to show you the kind of things that we are trying to achieve. Actually, these are 3 mice that were administered firefly luciferin RNA in one of our excipients for nasal delivery. So they got drops in the nasal cavity of the RNA in the formulation. And what you actually see is the RNA that has been translated, that got into the cell, that was translated into protein, and that lights up when you do the imaging. We're very happy with that picture for 2 reasons. First of all, it demonstrates that the RNA got into the nose and got transfected and translated. So it's actually a mechanistic proof that the delivery works. But also the fact that we managed to limit it only to the nose and that we don't see it dripping down into the intestinal tract or into the lungs is a big plus in terms of further development. So these are the type of data we're looking at today, and hopefully we can reproduce that in the next immunogenicity. The main question we're getting today, and I'm sure we'll get it during this webinar, and I'm getting it every day, is why do we think that it's worth to do this kind of different effort while there is such a many and such a big amount of big players moving very fast with more traditional and less risky vaccines? Now, we believe that there's a number of scenarios, and the base scenario, which we all hope will happen, is that by the time that we are ready with our product, there will be primary mass vaccination in place with an effective antibody. So where could we then fit in? I think we could fit in as a booster solution if we can demonstrate that our long-term protection is more stronger than just with an antibody-based vaccine. We could also fit in as an additional boost or an additional layer of protection for highly exposed individuals. Imagine in a local breakout situation, in a healthcare situation, where you want to offer people an additional support or an additional boost on top of the already administered vaccine. And then what we don't hope will happen, but what may happen, is that this solution could be used to ring vaccine outbreaks where we suspect a viral immune escape or a viral mutant at place. It could help to contain that new breakout and make sure it doesn't respread in the population. We also think that the nasal administration could help to reach coverage in certain regions of the world where it will be harder, because it's a much more convenient way, especially with self-administration, than an injectable vaccine. Now, in the worst case, which obviously nobody hopes for, it turns out that antibody-based vaccines are inadequate, that the strains aren't sufficiently covered, that the responses are too short-lasting, or that there are other phenomena in terms of safety and disease enhancement. But then obviously it's nice that there are alternative vaccines that are being developed in parallel and that could maybe replace some of the first-line vaccines. But let's not hope we go that direction. I think we're all better off if the first base case scenario happens. In all cases, we do think that this type of approach is a platform for future pandemic preparedness. That's also the case we're making to funding organizations like CEPI. We're not just positioning it as a solution for today, we're actually positioning it as a project that could fit into a global, more broader pandemic preparedness. So I'm already at my conclusion slide, so I don't think I'm going over time at this point. So conclusion, it's certainly a different, we think original, but still rational approach. It's not going to be the front runner. The eTheRNA vaccine is not going to be the first one to be available in the pharmacies. But we do believe that there's sufficient use cases to justify the effort. And whatever happens as a company and probably broader, we think that the insights from this program could generate new approaches to infectious disease vaccination in general, because we are actually exploring a new route of administration, a new way of using RNA, and a new way of actually making the immune response happen inside of the nose. And with that, I think I can conclude, and thank you very much for your attention.
Thank you, Wim, for this interesting presentation. So everybody who has questions for Wim or one of the following speakers, please enter them through the Q&A. Already 5 questions were entered, so please keep on doing that. And after the last speaker, we will come back to the Q&A part of this webinar. So now we move on to the next speaker, which is Professor Salens. So Professor Salens, you can start sharing your screen, or you're already doing that. The floor is yours.
Okay, good evening. Thank you for the invitation to the organizers, and thank you to the audience for joining this evening seminar series on COVID-19 drugs and vaccine candidates. So first of all, a brief recap of what coronaviruses actually represent. So these are a family of viruses that are very successful in nature. We can find isolates from different vertebrate species. These viruses also like us, and we are, for example, there are 2 seasonal alpha and 2 seasonal beta coronaviruses, which cause common cold-like symptoms, so not too much to worry about. However, as we all know, SARS and MERS, and then more recently SARS-CoV-2, cause much more severe lung disease with severe complications often, and unfortunately also mortality. And these have a zoonotic origin, and those are to worry about much more. So coronaviruses are enveloped, which means they have to at one point fuse their membrane of their virions with that of the host cell, and then infection starts. And for that fusion, they use quite complicated fusion proteins. These are the spikes that protrude from the virion surface. They are presented here from cryo-EM images from both MERS and SARS, complexed with FABs from human monoclonal antibodies. And these spikes, they are huge. They are the largest non-1 fusion proteins known to us. So they are 16 nanometer tall, protrude from the surface of the virion, which would be here. This would be the membrane of the virus. And we can divide them into those spikes into two domains, the S1, which is membrane distal, and the S2, which is membrane proximal. The S2 domain is responsible for the actual fusion, meaning it comprises the fusion peptide as well as the haploid repeats. And that is capped by the S1 domain. The S1 domain harbors the receptor binding functions. And this receptor binding domain is not always readily accessible in the spike when it is in a closed down conformation. So there is a oscillation going on, at least for SARS and MERS and for some other coronas, an oscillation where this the receptor binding domain, shown here complex with an antibody, FAP, can go into the up or down conformation. And this is the kind of sampling that the virus is trying to do to reach, to find its receptor, for example, ACE2 in case of SARS-CoV-1 and 2. And then the fusion process starts. This is a destabilizing activity that the protein undergoes. It also is an oscillation which at one point in time will expose the spike to antibodies which can, for example, neutralize, prevent the receptor binding domain from tackling its receptor, from engaging its receptor. Now, what I show here is the complex with FAPs of human antibodies published last year. Now, we were interested in our project in single domain antibodies, also known as nanobodies. And just a brief recap, this is what a conventional antibody looks like. It has a heavy and a light chain. It has obviously two antigen binding sites. However, in 1993, in the lab of Remo Hamers at the Free University of Brussels, they discovered that camelid species, such as the dromedare, have also these so-called heavy chain only antibodies, which differ in two aspects from the conventional ones. So first of all, there's no light chain. And secondly, the heavy chain is shortened. There is no CH1 domain available there. And the antigen binding site here shown in VHH is what we call an antibody, is the single domain antibody that we were looking after. In our case, for we and others published that these antibodies, these nanobodies, are interesting tools to target a number of viruses. For example, shown here for RSV, the pre-fusion form is RSV-F, which is complex with two nanobodies that we reported on a few years ago. The identity between, so these two proteins look very similar. Even though they look very similar, the identity in terms of amino acids is quite low. So they share only 29% sequence identity between the MERS and the SARS coronavirus. So we were interested in trying to find nanobodies which could potentially cross-react with the number of different spikes from different beta coronaviruses. Therefore, we envisioned back in 2016 this immunization scheme where we have a llama. One llama was chosen from a herd, and she was immunized with these different spike proteins each time. These are recombinant purified spikes from either SARS or MERS. And we came up with this regimen in an attempt to get cross-reactive nanobodies, which worked out partially, as I will explain. So after phage display and bioplanning, we indeed could isolate a number of nanobodies against the MERS spike, shown here in this ELISA, where we have nanobodies that bind the MERS receptor binding domain immobilized on the plate. This is the control, negative control. I think for SARS, we have two interesting nanobodies. Sorry, there are three shown here, which would bind to the RBD of the SARS-CoV-1 viral spike. These had a fairly high affinity for the target. So in an SPR assay, the MERS nanobody 55 had a 80 picomolar affinity for the RBD. The one against the SARS-CoV-1 had a somewhat lower affinity of about 1 nanomolar in this assay. So two nanobodies, or a number of nanobodies, which we picked out for further analysis. We picked out here nanobody 72 because it appeared to have cross-reactive aspects, cross-reactive activity. So then we moved on to determine the co-crystal structure of the, in this case, the MERS nanobody that we picked out. That is shown here in blue on the left, and complex with the MERS coronavirus receptor binding domain. And as you can see, it binds, it pinches with the CR2 and CR3, the receptor binding motif, where normally the DPP4 interface is localized, and therefore, obviously, in this model would clash with the binding DPP4, so it's in the way by binding to the receptor binding domain of MERS for DPP4 engagement. We have a detailed view on what's happening at the atomic level here. Now, the interesting one, or the peculiar aspect of the other nanobody, the nanobody 72 that binds to the SARS-CoV-1 RBD shown here in the crystal complex, this binds not to the ACE2 binding interface, which here sits like almost like a cradle ready to accept the ACE2 receptor. Rather, it binds to the site here of this protein, of this domain, and nevertheless, it clashes or it can hinder theoretically binding of ACE2 here shown in the in this complex by its CDRs, so by framework regions which are remote from CDR. So there's a clash over here, and this is actually even more dramatic if one takes into account this N-glycan glycosylation site here, which shows only the first proximal N-glycan residues on the ACE2 protein. So that glycan, which actually branches out quite heavily here in space, it clashes completely also with this nanobody 72. And again, we have a detailed view on the hydrogen bond interactions and as well as the Soldrich here on what's happening over here. So CDR2 and CDR3 are engaged, are important here in this binding. Now this site took our attention or caught our attention because this is again the SARS-CoV-1 receptor binding domain, where here we could have the ACE2 engagement. Here is the epitope. This epitope turns out to be very conserved across the SARS-CoV-1 known sequences. So there is some variation known there in the viruses that circulated back in 2002, 2003. But those variations, they are dispersed across the RBD away from the epitope. So the epitope remains conserved, hasn't changed in the SARS-CoV-1 viruses that circulated at the time. That was an interesting observation. So next we started to try to expand that conservedness or conservation across other beta coronaviruses. And here you can see a family tree of based on the spike RBDs of a number of SARS-like viruses. So SARS-CoV-1 I showed you. This is WIF. This is a bat coronavirus, which resembles very much a SARS coronavirus. And we took advantage of recombinant expression. And this is the RBD of, sorry, this is the superimposition of the SARS-CoV-1 RBD, where the green dots represent changes, differences between the WIF virus from bats and SARS-CoV-1. And again, we can see that the spot here where the epitope of SARS-CoV-2 is located is conserved and would normally not be hindered too much by these variations, which we showed in this SPR assay, so where we use immobilized nanobody and then flush over on the chip with the bat coronavirus derived, the bat virus derived RBD, and sure enough, we can see binding at the affinity of about 7 nanomolar. So it binds to another SARS-like virus that is circulating in bats. Here you can already see the SARS-CoV-2 popping up, which became apparent only earlier this year when it emerged in the population. It's related as well to the SARS-CoV-1 virus, as we all know by now. And we then also, of course, took advantage or wanted to see whether there was cross-reaction also with this RBD, and this we could demonstrate again in vitro with this SPR assay, where we have a measured affinity or a reduced affinity of about 40 nanomolar, which is again, due to a fairly fast off rate. So the binding on rate is high, but the nanobody dislocates quite rapidly in this assay. And nevertheless, we can explain this binding because, again, if we superimpose the SARS-CoV-1, SARS-CoV-2 changes, so in green we have the spots, the amino acid residues that are different between the two viruses on the RBD. Most of them are not changed at all at the level where the epitope here is all the SARS VHH-72. The clashes that I proposed or showed in theory with the co-crystal structures were also looked at in this assay, where we look at competition between the ACE2 binding for SARS-CoV-1 and SARS-CoV-2 with or without an antibodies. And this is just to show you that for SARS-CoV-2, here the line in red we see in this biolayer interferometry assay that as ACE2 is mixed or when the RBD is mixed with ACE2, we see a signal popping up. So there's an ACE2 engagement by the SARS-CoV-2 RBD. However, in the presence of nanobody 72, here shown in the black line, that signal is almost going back to zero, suggesting or indicating that there is competition in the presence of nanobody 72, so that nanobody 72 prevents the ACE2 engagement. The same is seen for SARS-CoV-1, going from the blue line here to the purple one, and then also for MERS. And we use the respective nanobodies as negative controls for these assays. So there's competition between the ACE2 binding of SARS-CoV-1 and SARS-CoV-2 and VHH-72, so the VHH can prevent that binding. So then in terms of infectivity, of course, this is all very nice biochemical data and structural insights that we gained. And then we were wondering whether there was any effect on the
Jongens, je moet zelfs wel stilletjes zijn, want ik moet zelfs terugspreken.
I beg your pardon? So here we look at the neutralization of a so-called VSV pseudotype virus. So these are vesicular stomatitis viruses that have spikes coming from either SARS-CoV-1 or SARS-CoV-2, and they report infection or entry, I should say, by a luciferase signal. And here you can see that dose dependently, the nanobody 72, just this as an example, is able to prevent that entry of the pseudotype virus for SARS-CoV-1, which we would expect because that's where we selected the nanobody against. This is the other one. Controls are fine. And then for SARS-CoV-2, we see also a moderate, but nevertheless direct, quite clearly measurable effect with the nanobody 72, where the nanobody 6, which works out here fine, doesn't do anything against SARS-CoV-2. So there is evidence of prevention of entry of either SARS-CoV-1 and SARS-CoV-2. Now, this entry prevention, this neutralization of the pseudotype viruses is much more pronounced when we start to make fusions between the nanobody and the human FC protein domain. So this is a recombinant nanobody FC fusion, where the nanobody 72 and not the controls clearly can suppress dose-dependently the entry of the SARS-CoV-1 as well as the SARS-CoV-2 pseudotype viruses. We also took or made efforts to express the nanobody FC in a CHO cell system, which is a comforting expression system for pharmaceutical companies. So it's a workhorse for recombinant protein expression and biologics that are in clinical use, especially for antibodies as well. So this is CHO-produced SARS VHH-72, so the molecule shown here that was produced in egg cells initially. So this is now in the robust CHO cell system, and you can see here in the RBD assay that the binding is much more, has a much stronger affinity, especially the off rate here is largely much more reduced. So the off rate is clearly reduced with an nanobody FC fusion. This Homeostasis gel here shows that if we express the nanobody FC and harvest the cells in CHO, these are ex vivo cells, and harvest those cells, the medium from those cells at different times, without any concentration, we pick up clear bands of expressed nanobody FC. We easily reach here 300 to 500 milligrams per liter in just shake flask in the lab environment, so without any optimization whatsoever. And this actually beats or is in line with the control antibody. This is a licensed monoclonal against CD20. So that is also produced in the same cell system, and this clearly not performing better, rather the contrary than an antibody FC. And again, this CHO-produced material purified from the cells nicely prevents SARS-CoV-2 VSV pseudotype viruses from entering. This is the antibody FC control, which doesn't work, and this is our negative control. So the CHO-produced, ex vivo produced nanobody FC is capable of neutralizing SARS-CoV-2 pseudotypes, and is also expressed at substantial amounts, even in the lab environment. So then all this is still nice in vitro data on binding and neutralization. And eventually we could do an experiment thanks to the collaboration with Johan Nates and his team, who explained last week, his mouse and especially the hamster model for SARS-CoV-2. So these animals are experimentally permissive for SARS-CoV-2 infection. And what we did was Johan and his team did was to infect the hamsters on the day. So prior to infection, they were injected intraperitoneally with nanobody FC or human serum as a control. And the next day they were challenged with 10 to the 5 TCID50 of the SARS-CoV-2 virus. And then on day 4 after the challenge, blood was taken, but also the lungs were homogenized to look at the viral loads in those lung homogeneous. And what you can see here and appreciate from the graph is this is the there's a relevant control setting where we use actually synergies instead of the non-antibody RC. So that gives us a certain level of RNA of the virus. These are the human convalescent serum. This is a convalescent serum. This is a negative control serum, but that didn't work out too well in this experiment. However, with the non-antibody RC treated animals, we can see that all 4 of them have up to 100,000-fold reduction in viral loads based on RNA measuring here. So it works, at least in vivo, also in this experimental model in a prophylactic setting. And this brings me to the conclusion. So the work, except for the hamster work, was published in May. You can read all about it if you want to do so. Conclusions are that MERS-CoV, SARS-CoV-1 and SARS-CoV-2 neutralizing single domain antibodies were isolated. Please keep in mind that the MERS coronavirus antibody does not cross-neutralize the SARS-CoV-1 and 2 and vice versa, but the SARS-CoV-1 and 2 are cross-neutralizing. We obtained high-resolution structural insights, which reveal the mechanism of action, which is occluding of the ACE2 binding site in case of the SARS viruses, and also probably destabilizing the fusion proteins before they can engage the receptor. And then the SARS VHH-72 domain construct or fusion construct is, in our opinion, a promising candidate for further clinical development, for example, as a prophylactic or a therapeutic intervention measure against COVID-19. Now, this was a team's effort, and I would like to hugely thank people from our center and Doreen Bert in my group, and then Nico Kalwaard, him personally, for his drive in getting the COVID-19 work at full speed running in these difficult times early this year. And people from his group, Jason and Daniel, helped with the co-crystal work. Barney and Kizzy with some neutralization work early on. We got reagents from a team from a group in Germany. Reza, huge thanks to him for the non-antibody surface facility work, who organized the immunization of the llama and the phages play library. I should also thank people in Riga listed because they helped us out with this hamster model in difficult times because they were over-demanded to service their hamsters for all kind of parties, I suppose. So thanks to that, field discovery sciences and knowledge transfer are extremely important also to get this product a step further from a paper and so into a clinical path, which we hope to elaborate later on this year. And with that, I end with this people involved in our center pictures here somewhere in March or April this year. And I would like to thank you for your attention.
Thank you, Professor Salens. Then we move on to the third speaker of this webinar. So Professor Lambrecht, can you start sharing your screen, and the floor is yours.
Okay, thank you very much. I would like to talk about the use of anti-rheumatic drugs for the treatment of severe COVID-19 infection. Okay, so this scheme is actually, I think, one of the best schemes that certainly is used in the clinic a lot. And it shows you that there are various phases to COVID-19 infection. So first phase of infection is mainly characterized by a viral response phase, where there's a lot of constitutional symptoms, fever, some diarrhea, sometimes headache, typically flu-like symptoms. And in the majority of patients, this will actually go away. But we see that in about 20% of patients, there is a second phase of the disease where increasingly you get pulmonary symptoms. So you see here phase 2A, phase 2B is characterized by pulmonary phase where there's increasing shortness of breath, hypoxia, and there's abnormal chest CT and some abnormalities in the liver. And this can go from worse to worse. Phase 2B is really when you start getting almost respiratory failure, where you are dependent on oxygen. And then in a further minority of patients, I would say around 5% in total of patients, maybe even less, maybe 2 to 5% of people, you will have this hyperinflammatory phase where people will end up in the ICU on mechanical ventilation. And this stage of the disease, there's often ARDS or acute respiratory distress syndrome, and even things like shock and cardiac failure can occur. So the various drugs that have been used and that we have heard about so much, like chloroquine, remdesivir, convalescent plasma, they have basically been used a little bit across the board, and that has led to very, very confusing findings. The viral response phase, this is where viral replication is the highest. This is actually where you should be using an antiviral drug. Whereas in the later phases of disease, where there is pulmonary symptoms, hypoxia, and ARDS, there's more and more evidence that in fact it's an overt or an overzealous reaction of the immune system, so by the host inflammatory response, that is responsible for the symptoms. So it is less likely that in this stage of the disease, when people are already in the ICU, that an antiviral drug will do very much. So this scheme actually was very helpful to us. We used it already in March, I would say, to position our clinical approach. So to bring it to the idea of using antirheumatic drugs, we have to understand a little bit how the lung is actually working. And in fact, a lung, the primary function of a lung is to exchange oxygen. And this oxygen and CO2 exchange, this gas exchange, is occurring across the alveolo-capillary membrane, which is an extremely thin membrane of only 0.3 micrometers thick. And you need this short distance because otherwise the gases cannot diffuse. But you can also imagine that such a short distance, it's very easy for a pathogen that would come in here in the alveolar space, say like a gram-negative or a gram-positive. It's very easy to get through that barrier and become entrapped in the bloodstream and lead to a systemic infection. So this is the big problem of the lung. So on the one hand, you need to maintain gas exchange. And on the other hand, you need to defend yourself against pathogen entry. So normally you do this by mounting an inflammatory response and sending immune cells into the tissues. But you cannot do that too much because otherwise you're going to compromise the gas exchange. If you would fill this up with inflammatory cells here, this barrier will thicken up and there's no oxygen exchange anymore. And this is why we have alveolar macrophages in our lung. Alveolar macrophages are immune cells that can actually handle a lot of inhaled pathogens. They can get rid of them without causing a lot of inflammation. I always call them the vacuum cleaners of the lung. They just take rid of the pathogens and they avoid inflammation. But they do other important things. They are also important for surfactant homeostasis, so they keep the lungs in an inflated, open condition, and they keep T cells in check. So a lot of T cells in the lungs are actually in an anergic state. So if you're thinking about generating a vaccine that will generate T cells that go to the lungs, often in the lungs, those T cells will be kept in quiescence by anti-inflammatory alveolar macrophages. So what happens in early infection? So early infection, the virus infects type 1 and type 2 alveolar epithelial cells. So very deep in the lungs, you get infection. The alveolar macrophages disappear. So that is very striking in this COVID-19 infection. You get a complete disappearance of alveolar macrophages. So the vacuum cleaners go away. So immunoregulation goes away in the lungs. And then there's also through the barrier, there's infection of endothelial cells that get infected. So initially the immune system will react to this, and the immune system will react by producing cytokines. And cytokines are initially very good. So one of the first cytokines that is produced is type 1 interferons, and these will, coming from the lung epithelial cells and from alveolar macrophages, will actually cause a so-called antiviral state in the vicinity. And it's been thought of that people of advanced age, so elderly people, that they might have a defect in type 1 interferon production. So they don't produce it quickly enough, so the virus has more chance of replicating. But you also see that many of these cytokines here that are produced locally, either by lung epithelial cells or by macrophages upon early infection, get triggered to TLR7 agonism, but many of those cytokines are in fact very, very good for promoting anti-defense mechanisms. So a little bit of neutrophils coming in is very good, a little bit of monocytes coming in is good, because in this way you can fight the infection. Many of those cytokines you will recognize immediately, TNF-alpha, IL-1 and IL-6, are typically targeted in rheumatoid arthritis. So early, if you would do this early in the disease, this would be a very bad thing. So if you're going to give an anti-rheumatic drug in the viral phase of the disease, let's say a couple of days into infection, this is actually going to be detrimental, because you need those defense mechanisms early in the disease. So this is an example of a patient where you see, this is the 5th of April, was in a referring hospital. You see here there's some ground glass opacities. This is a very mild disease. Patient was admitted for a couple of days, and then 5 days later, same patient. This goes completely wrong. You see here that the disease has spread all over the lungs. This bilateral white lungs in just 5 days time. So this patient has progressed from this phase of the disease, where there is no hypoxia, all of a sudden to this phase of the disease, at the borderline of going to the ICU, where you can expect the host inflammation to be overreacting. So how does this look like? In this stage of the disease, when you're seeing widening out of the lungs, you will actually see that there is a lot of capillary leak happening. There is overproduction of bradykinin, because the ACE2 is functionally inactivated through the virus infection. And you will actually see that bradykinin contracts the lung endothelial cells so that they become leaky, and you get really flooding of the alveolar spaces with fluid. And you will see the formation of microthrombi. It's very typical of the disease. You see capillary microthrombi very, very early when you get this lung injury response. And this goes from worse to worse. So the microthrombi will actually start extending into the tissues. There's tissue factor formation. You will see fibrin deposition, not just in the thrombi here, but also in the lung alveoli. And this will be rapidly broken down by plasmin, leading to the occurrence of D-dimers. So D-dimers are very important biomarkers that are often used in the rheumatoid studies or the anti-rheumatoid drug studies, because D-dimers, when they start rising, it's actually a sign that things are going really wrong in your lungs. This is called hyaline membrane formation because this proteaceous material covers the basement membrane, and it really causes a very, very severe problem, because the epithelial cells cannot recover anymore when this is covered by fibrin. So then it goes from worse to worse further. Remember, in this disease, CRP is a very important biomarker. So CRP, we normally see it only with bacterial infection, but this is a viral infection where you get tons of CRP, and the CRP can deposit in the lungs, and there's evidence from MERS infection that this is happening. Also in SARS, it's been shown already. So CRP gets deposited in the lungs, often on the dead cells and on the hyaline membranes, can activate via the classical route. It can activate C3 and C5 complement, and complement through the membrane attack complex can further kill lung epithelial cells, and it can further cause microthrombi. And C5 is a very well-known cause of microthrombus formation. So is the membrane attack complex. And breakdown products of C3 and C5 are C3a and C5a. These are chemotactic factors, and they will actually recruit more inflammatory cells, more than normal, more than what you need for that phase of the disease, will be recruited by this complement activation cascade. And the cells that flux into the lungs are in fact the cause of the so-called cytokine storm. So it's a very atypical cytokine storm, because in most of the cytokine storms we know from the literature, the primary problem is in fact in the T cells. Think about CAR T cell therapy. where you will have T cells reacting to a single antigen. They can produce tons of GM-CSF, for example, that can cause cytokine storm and activation of downstream cytokines. Here, the cytokine storm is not coming from the T cells, it's mainly coming from the monocytes that are recruited into the lungs in response to this complement activation, in response to this damage signal. And again, we see the same cytokines that we see early in the disease now start playing a detrimental role. IL-1, IL-6, and TNF-alpha are very much implicated there. And this is just some data from our own clinical studies that we did in Ghent and in Bruges. It's a so-called SARPAC trial where we're looking at an inhaled cytokine, sargramostim. So here you see the levels of IL-6 in the serum of a cohort of age-matched healthy controls. And this is the levels of interleukin-6 in COVID-19 patients at day 1 of disease, day 1 of admission in hospital, I would say, and here day 6 of the disease, where most of them would be around day 14 in their disease process. And you clearly see that IL-6 is increased compared with healthy controls, but this is getting better already in the majority of patients after 6 days of hospital admission and treatment with our various strategies. So interleukin-6 is certainly enhanced in the serum and in the bronchoalveolar lavage fluid of patients with COVID-19. So this is a patient who unfortunately died in our hospital. So this was a young person, I think it was a 55-year-old person who died of severe ARDS. And this is just an illustrative picture here that I got from the pathology department from Jo van Dorpen, where you actually see a staining for CD163. So CD163 is normally, it's an activation marker on macrophages that eat other immune cells. So for hemophagocytic macrophages are CD163 positive. And you see here that all the alveoli that are still left are actually filled with these CD163 positive mononuclear cells that can eat other immune cells. So this kind of becomes a graveyard for your immune system, because every immune cell that gets in there, like your lymphocytes, would be eaten by those macrophages. It's exactly what we see. A lot of those patients have lymphopenia and have eosinopenia, and the thoughts are that those cells are killed in the lungs, and at the same time those cells will produce interleukin-6. So this is a bit of an illustrative case. This is what we are doing for the moment, also in our clinical trials. We are using very advanced technologies like single-cell RNA sequencing on lavage fluid in collaboration with Linus van der Kerkhof in the COVIM trial. We're also doing this in our clinical trials with biologicals. We're actually sampling both the blood as well as the alveolar compartment. And then we're doing single-cell RNA sequencing. And then we allow a very clever clustering algorithm developed by Ivan Sais at VIB to cluster your cells. And you see immediately here that the majority of cells here are mononuclear cells. And some of those mononuclear cells are pro-inflammatory or pro-fibrotic monocytes already. We have some HLA-DR low monocytes, which are typically seen in sepsis. We see subtypes of dendritic cells here, very few basophils. And then we also have a lot of T cells and NK cells and some mate cells. But these very detailed analysis actually allow us to zoom in on which cell types are there, which cytokines and factors are they producing. I'm just showing you, like, for example, here, this cell type here, the FCN positive monocytes, pro-fibrotic monocytes. It's just the top 10 of genes here, but we can also do this for a top 200 genes. So we can even do pathway analysis on individual clusters of single cell clusters to give you an idea of what's happening in this disease. And just as an illustration here, all the mononuclear cells that we see here influxing into the lung. So this entire cluster here, if you look at the expression of C5a receptor, for example, this is the receptor for chemokines, for complement C5a, you see that those myeloid cells are hugely expressing C5a receptor. And this is also the basis of a clinical trial we're doing with zilucoplan, a drug from UCB that blocks C5 in COVID-19. So, and then the last phase of the disease is in fact the phase we want to avoid, is when there is too much damage to the lungs and the fibrin cannot be cleared because the alveolar macrophages are gone and they cannot clear the debris. What you will actually start seeing is that the lung epithelial cells try to repair the lung. They start going into overdrive, they go into hyperplasia, and hyperplastic type 2 alveolar epithelial cells make two big things. They make TGF-beta and they make EGF, so epidermal growth factor, and they make transforming growth factor beta. So what will happen is that you will get thickening of the septa with fibroblasts, and those fibroblasts will also start proliferating, and at a certain point they have nowhere to go but to go into the alveolar spaces. So this is drawn here. So this is a region where the fibroblasts actually go into the alveolar spaces and completely fill up the alveolar space. And this pattern is called cryptogenic organizing pneumonia, and it's a cocktail for later fibrosis. This aspect of the disease you want to avoid, but initially it's still steroid sensitive. If you wait too long, this becomes steroid insensitive and you end up with irreversible fibrosis. So this is again an illustrative case from a patient who died in our hospital where we did an autopsy. And you see here, this is an alveolus here. You hardly recognize it as an alveolus. This is the interstitium, which is completely thickened. And you see here that all those epithelial cells are actually cuboid type 2 epithelial cells. And because these guys make EGF growth factor, so epidermal growth factor, they actually transdifferentiate into squamous epithelial cells and they start making keratin. So this is called squamous metaplasia. It's very irreversible and it's a sign of severe deranged lung repair mechanisms. And this is again, it's the same patient, another region of the lung. This is the healthy control here where you see the normal alveoli in the lungs. And you see this patient who has this squamous metaplasia, you see that these alveoli here are completely filled with young islands of fibroblasts. So the fibroblasts have broken through the alveolar spaces and they're actually filling up the alveolar spaces. So all of this, it's one giant ball of fibroblasts making collagen. So you better give some steroids here early, you know, because in a couple of days this is irreversible and it starts retracting and you get irreversible fibrosis. So in the end, if you're quick enough and you can halt the damage pathway, you can halt the inflammation, you can get rid of those monocytes. Actually, the monocytes can differentiate into alveolar macrophages again. The recruited monocytes, they do this under the influence of GM-CSF. And then those monocytes become alveolar macrophages that become vacuum cleaners again, and they can still completely repair the lung. This is what we're aiming for, and we're seeing the first patients now 12 weeks after disease. We do have patients where they had a complete white lung, and you see them back 12 weeks later, and their lungs are completely healed. We don't see anything anymore. We unfortunately, however, also see patients where we already see bronchiectasis and where we see the beginning of subpleural fibrosis, which is the beginning of lung fibrosis. So this brings me to the final set of slides. So we have set up in Belgium, we have set up a trial called the COVID trial, which is the coronavirus anti-interleukin drug trial. The trial has been set up in collaboration with KCE, so the Knowledge Center for Drug Use. It's a government organization with Frank Hulstad has been instrumental in driving that from KCE. And what we're actually doing in this trial, and it's quite unique compared with the other trials that are currently running around anti-rheumatoid drugs, is that we are comparing not just, most of the trials are either using IL-1 blockade or IL-6 blockade or TNF blockade. Most of them are actually around IL-6 or around IL-6 receptor. What we are doing is we are comparing the effect of IL-1 blockade with the IL-6 blockade. And within the IL-6 arm, we have two strategies to block IL-6. We have a group of people where we block with anti-IL-6 antibodies, siltuximab, or we are blocking with an anti-IL-6 receptor antibody, tocilizumab. So this makes for 6 groups of patients, so usual care, and this includes dexamethasone steroids, that is allowed, anakinra alone, an IL-1 blockade, and then siltuximab alone, anakinra plus siltuximab, tocilizumab alone, or anakinra plus tocilizumab. And because this is an adaptive block design trial, we can actually compare the effect of IL-6 blockade with IL-1 blockade, and we can also compare the effect of the combinations. And so this is the effect of IL-6, sorry, and this is the effect of IL-1 blockade. So the trial launched a little bit when the peak was starting. Well, it was really actually just over the peak. You have to think of this. We had to bring this protocol to the FACH. We had to get ethical approval, design the entire protocol. I think we pulled it off in something like 14, 15 days, which is something that normally takes a couple of months. And on top of that, we also had to have the approval of 16 centers in Belgium. So nevertheless, we did this in a couple of two weeks, I would say. And then the epidemic started going down. But currently, there are 88 patients enrolled in the trial. But we decided to keep the trial active. So rather than breaking it open now and not be able to conclude anything, we don't want to hurry. We know already from the countries around us that IL-6 blockade seems to work with siltuximab and with tocilizumab. There's a lot of reports from France and from the US going along those lines. We also know that IL-1 blockade is doing something, but this trial is unique because we will be able to compare the synergistic effect and also the IL-6 versus IL-6 receptor blockade effect. So that's why we decided to go on with it and just prolong the accrual period. So I'll just briefly go into what type of patients we're going for. These are patients who are in phase 2B of the disease, so they have severe hypoxia. So typically, even if they get oxygen, their saturation is below 93%, or this is very technical. This is the PF ratio. It's a typical ICU measurement that can also be done on the wards. It's arterial pressure of oxygen divided by the percentage of oxygen in the air. If this is below 350, they are eligible. They have to have bilateral infiltrates on the CT scans. They have to be beyond the viral phase already, so minimum of 6 days of symptoms, up until 16 days of symptoms. We don't want to be too late either. And then they have to have signs of a cytokine storm. A lot of trials around us, and also in the US and in China, have not randomized their patients or have not used inclusion criteria for cytokine storm in their inclusion. They just took people on the ICU, assuming they would have a cytokine storm. And we have seen that that's not the case in every patient. So we are screening patients with high ferritins, and particularly ferritins that are also rising. It makes no point to treat a patient where the cytokine storm is already going away. So we are taking people in the rising part of the ferritin curve, people who are lymphopenic and have other signs of cytokine hyperactivation syndrome, like increasing LDH, CRP, ferritin levels that are increasing, and D-dimers that are increasing. And we're measuring clinical responses, ICU need, survival. There's like a list of 30 things we're measuring, oxygenation parameters, but also very uniquely, we are also looking later in the disease whether we can prevent fibrosis. So this phase I was describing leads to fibrosis. You can measure this with lung function. So we're seeing those patients back 20 weeks after their episode. And also quite uniquely, all the things I was showing you there, the endothelial damage, the fibrosis damage, we're measuring all kinds of biomarkers that are made by those hyperplastic type 2 pneumocytes. We're measuring them in the serum, and we're combining this with a really very expensive tool, which is single-cell RNA sequencing on blood in all those patients. So you have to imagine this is 340 patients, very expensive. And then also with CytSec CytSec technology, which is a combination of single-cell RNA sequencing and barcoded antibodies, so that we can come up with better flow panels in the future. So the only thing we're very careful about is that we're not causing secondary bacterial infection. Think about this role of cytokines in the early defense against pathogens. We want to be sure, that with IL-6 blockade, we're not causing fungal infection, for example, or other infections. So we're monitoring this with a very low threshold, and we're taking along procalcitonin levels, for example, as part of routine follow-up of those patients. Okay, the only one slide I want to show here is that this is only possible, with a dedicated team of doctors. So these were 5 VIB doctors, 6, well, I'm here actually, 5 VIB doctors who were working on various immune projects on lungs, who dropped everything. They were all medics, and they started working full-time on these trials. This is the clinical trial nurses who are involved in this, and all of this would not have been possible without KCE, Frank Hulster, Nelle Stokar, and then the HIRUS team, which is the clinical trial unit at UZ, who is setting up all these things for us as a service. So thanks for your attention.
Okay, thank you, Professor Lambrecht, for this interesting presentation. So now we can go over to Dr. Jamila Alouahed, VP and Head of R&D Center Belgium, GSK. So if she can start sharing her presentation, she can start.
Immune response in pathogens. So if I can summarize the technology wave into the vaccine field, starting from a whole virus, inactivated or attenuated vaccines to subunit vaccines, and now we are moving to new technology that we heard earlier on, like RNA technologies. We see also that not one technology can fit all vaccine demands. That's something that's also very important. And also we see that infectious disease can have a different mechanism according to the age of the patients. And one of the important knowledge in the adjuvant portfolio is exactly what we are trying to do, is how can we use this immunoenhancer to induce the right immune response according to the age populations. Again, if I pick up one example, recently we have been able to launch vaccines against shingle disease in elderly patients with an unprecedented active efficacy in these age populations, up to 19% of efficacy. And I hope that in the coming months, in the coming year, we have on this road, which is COVID-19 vaccines that will be part of our innovation. So as I said, our discovery engine are based in Rensensar. And one important element is that most of our vaccine pipeline has been done through collaboration. And you see here a quite high number, more than 90% of the vaccine that GSK has developed were done through collaboration with partners around the world. And we have used exactly the same approach to tackle the important need and race to have a solution for COVID-19. And the way we have approached this challenge is, on one hand, can we leverage the expertise that we have in-house in terms of understanding what are the needs of having a successful vaccines, having the tools to be able to scale up, because we are talking about a world need, and also how can we leverage this partnership to get faster and have the increased chance to deliver vaccines. Technology platforms are very important, and as I mentioned, not one technology can fit all the needs in the vaccinology field, and you have here a toolbox that GSK has been able to gather through the years that was associated to development of a particular vaccines with the aim to ensure appropriate efficacy. Reverse vaccinology has been used for MenB. We are using the adenovector platform that you have heard that is being used by our colleague in the RSV pediatric field. and it is used for COVID-19 by J&J and AstraZeneca. Bioconjugation, and we have talked about mRNA that we also have in our toolbox. I would like to focus more today on why we believe that the adjuvant system are important in potentially identify and develop a vaccine that would be efficacious against coronavirus. So first, few words about adjuvant. So what are the adjuvants? Those are essentially important when you have subunit vaccines. They allow to have the right environment in terms of presenting the antigens once the vaccine is injected to induce a strong immune response, antibodies, but also ensuring that we create the immunologic space for a memory immune response and have translated by a high efficacy. We speak about adjuvant system quite often, and if you have here a schematic view of what we have in our adjuvant system, is on one hand the type of formulations, which could be liposome, all in water, or aluminium, mixed with immunoenhancer, and the combination will provide you the adjuvant system that is being used in several vaccines pipeline. What we are proposing is to use the middle one for the COVID-19, which is a pandemic adjuvant that has been several times in clinical, but also has been used in pandemic H1N1 flu, for which we know how to produce it. We have safe databases into this adjuvant, and also, more importantly, the process for manufacturing this adjuvant. Few data on this pandemic adjuvant. This is a clinical study that has been done in collaboration with Professor Leroux, where we have compared in populations that are young and elderly patients, the H1N1 vaccines, plus minus the antigen, the adjuvant, and you see also a dose range. The take-home message is that using the adjuvants, you not only increase the number of subjects that will seroconvert after vaccinations, but also you can use up to 10 times less antigens, which is critical when you are in a pandemic, as we have to increase the supplies. So that's our really insight that was very important in the context of the H1N1 pandemic, that we believe are also key in the current COVID-19 development. So summarizing here the value of the adjuvant, so the antigens, and we'll come back on what are the antigens that are now used for those developments, combining with the adjuvant to increase the immune response, which has will allow to have a strong immune response and use of less antigens. I'd like also to emphasize, as we have heard, the target population that are the more sensitive to the COVID-19 are elderly patients. And we know that those patients have what we call immunosenescence. Their immune system are less resistant or less robust than young people or young adults. And we have seen that adjuvant can not only induce a strong immune response, but can also make the difference in the elderly populations. And today, the challenge you are having in the vaccine for COVID-19 is on one hand, have an efficacious vaccine, a vaccine that can be scalable, but also a vaccine where the efficacy would be also seen in young adults, but also in elderly patients. That's why we do believe that having an adjuvant is also part of the success of potential vaccine in the future. So I have spoken about the adjuvant, which is one part of the vaccines, and we have collaborated through the world with key partners that have technology, had science that were already in place in the field of coronavirus, to really leverage their expertise with our expertise to develop a vaccine. And you see that you have here several partners with whom we are collaborating, and with whom we are providing the adjuvants to do their preclinical. And I'm happy to announce that the first clinical study has started 2 weeks ago in Australia, in collaboration with Clover. Unprecedented crisis has led also to unprecedented collaborations. We have also looked at what could be the best way to ensure that we have not only the opportunity to maximize the expertise of leading vaccine companies, but also making sure that we can leverage also the expertise in manufacturing to be able to produce sufficient vaccine down the road. And we have announced the collaboration with Sanofi, where Sanofi will use their expertise that has been built in the flu vaccine to deliver primary S1 antigens, and together with the combination with the adjuvant from GSK, we'll move into the clinical or hopefully have something that will function. So if successful and with regulatory considerations, we aim to have a vaccine that could be available by the end of next year. So Sanofi, Clover, that's not all the collaboration that we are having in place. Other will soon be in the clinical. And you have here a snapshot of the one that are more advanced. Most of them are using the S1 trimeric protein as antigens with different type of technology to stabilize the trimer, which we know is critical to ensure that the scalability and the reproducibility of the productions will be important. And we have one collaborator which are using another part of the virus, which is the receptor binding domain. And hopefully having this type of collaborations allow not only to increase the potential to have a highly efficacious vaccines, but also will allow to provide sufficient vaccines for the world, as all those companies will be also able to produce the vaccines. So most of them should be into the clinic by the end of the year. So our approach was really to ensure that we leverage the partnership, and we have committed to deliver the necessary needed adjuvant. In addition to what we had in the preparedness of any pandemic in flu, we have started to produce 1 billion of doses of adjuvant in the future. We are really looking for a global approach, as I have mentioned with different collaborations. And also, we hear a lot about who should be having access, and we will continue the philosophy that we have used for many other vaccines. We are committing to provide our adjuvant and vaccines to the world in collaboration with the global health governments. And also, the current pandemic is teaching us something that GSK has been very vocal the last decade about being prepared for pandemic. We have seen how much effort we need to be put in place to get in the race, but also to have the structure for manufacturing in place. And GSK has been in the wave of the previous epidemic, and we know how consuming it can be. So today, the insight we are having from the COVID-19, there is a clear understanding that we all have a play in making sure that we are getting ready for the next pandemic. So everything that will be put in place for COVID-19 will further be expanded to get ready for the future. So the access is an important also element. You know that GSK produce 70% of the vaccine that GSK produce go to developing countries, and we want to make sure that people from those countries will not be left out from COVID-19 vaccines. And as part of our commitment, we will be giving donation of our adjuvant. Last but not least, we need to be fair. We have squeezed the development of vaccine that often take in average 10 to 15 years into a development which is less than 2 years, which is unprecedented. And the reason to believe that we can do it is, first of all, the science behind coronavirus was known through the MERS and SARS, so which has allowed to have quite rapidly candidate antigens to move into the clinical. But there is also a partnership from all the stakeholders that is allowing to accelerate. But we know it will take time to generate the data, and it is important to ensure that meanwhile, we have also alternative solutions that can either prevent acute disease or having alternative way to prevent or mild disease. We have put in place also a collaboration with a company called VIR, where we are moving a monoclonal antibody against COVID-19, and this should be in the clinic by the next few months, which also would provide additional solutions to be able to have alternative while we have a vaccine that is available. Last but not least, we are also, as many other companies, try to repurpose some of the molecules that are being developed in other viral disease or any inflammatory molecules. We have heard about the impact of the anti-IL-6. We are also evaluating anti-GM-CSF molecule through our pharma colleague, and we are looking also at repurposing some of the antiviral medicines that has been developed by the VIV company, so antiviral developed for HIV disease. So all this really help us to ensure that there are alternative treatments that can be available for patients that will acquire infections, while we are much more mature into the development the vaccine solution. And I'd like to finish there, and I think this is really unprecedented and show also the value of all the effort that has been done in the vaccine field to better understand the immunology, the interplay between the infectious agents and the host. And you have heard beautifully from Bart that it is important that you understand the immunoinflammatory molecules that can be triggered by those pathogens to have solutions not only to treat, but also to prevent. And all those learning will clearly help us in the future to tackle other infectious agents. So I'd like to end here, and thanks for listening to me.
Thank you, Dr. Jamila. We would like now, in sake of time, go straight on to the Q&A. And we have one last question for every speaker. So the last question for Wim is, during the vaccine development, how is it taken into account that the coronavirus is known to mutate?
But actually, that is one of the features that we're trying to build into the vaccine. When we do the selection of the epitopes, CD4, CD8 epitopes that will be part of our construct, we are actually looking historically, obviously. We don't have a crystal ball to see the mutations of the future, but we're at least looking within the corona family as we know it, which stretches and which epitopes could give us the best cross protection. And we hope that if a next mutant comes along, that at least it falls within that family. If it's not the case, I think we're all in trouble anyway. Most of the vaccines, I think then the game is to be able to switch over as fast as possible to a new antigen, to a new construct that again can protect against. And that's why developing a platform like many of the speakers today talked about, I think is critically important.
Okay. One question to Professor Salens. It seems unlikely that viral infection still plays an important role in the severe inflammation phase. So how will you decide which patients should be treated with the medication during the viral phase?
Yeah, so the first trial will likely be organized in patients who are diagnosed early after infection, not yet hospitalized, or possibly just hospitalized with mild symptoms. However, I'm not totally convinced that the virus does not play a role even at the late stage of disease, because if one looks at the occasional convalescent serum transfers where probably the neutralizing antibodies played a role, in many cases severely ill patients were recovering. So that might be in a later, that those patients might also be considered if early stage trials are going fine.
Okay, one more question maybe for you was written in the Q&A by Antonis Ficatas. The question is, have you also tried to evaluate the RNA levels in other organs except lungs and mice?
Yeah, so we haven't looked in mice, but rather in hamsters for now. We've looked in the ileum as well, and there we see a reduction too. In the throat swabs of the hamster, so this is a very acute, rapidly progressing infection in those animals. In the throat swabs, we saw only a modest reduction. So the ileum was reduced as well as were the lungs.
Okay, and maybe one last question for you. There is it was put in the Q&A anonymously. The question is, what is the anticipated treatment regimen for neutralizing antibodies or nanobodies? Does it require chronic treatment to prevent and cure? And how is the immune response impacted?
Yeah, that is a complicated question. So the nanobody Fc fusion that we have developed is will likely have a fairly long half-life in circulation. So one could infuse the antibody construct and probably count on a few weeks of protection, or at least availability of the antibody construct. So chronically, I mean, it would likely not require a repeated treatment every other day or so. With regard to the inflammatory response, or the immune response, this may relate to the possible recognition of the drug as a foreign antigen. So there might be anti-drug antibodies developed after some time. So this we cannot exclude.
Okay, thank you. One last question for Professor Lambrecht. Many patients with moderate disease report persistent symptoms of, amongst others, fatigue and reduced capacity. How many patients do you anticipate that will end up with severely reduced lung function after COVID? And do you think that the anti-inflammatory treatments will be able to counter lung fibrosis?
Okay, those are excellent questions. So it's also the reason why we're doing the trials. No, it's to look at the long-term effect on fibrosis. So in terms of patients feeling, feeling tired and exhausted and headaches after disease, this is not so uncommon after a viral disease. We see it with every severe infection. If you have an EBV infection, you can also be really tired for months. I think it's too early to say now that this could be a trigger, for a chronic fatigue syndrome or something, or a long-term impact on fatigue. The disease is just too young for that. However, we must take into account that there is some neurotropism for the virus. There are some reports that there is really encephalitis in some patients. In the patients that we have done autopsies in, there were signs indeed of neural involvement. So, we have to be vigilant, I think, that there might be some chronic effect on the brain, but it's too early to say. In terms of fibrosis of the lung, if you see those white lungs and you look, for example, in ARDS, where you also see those white lungs, normally it's around, I would say it's below 20% of patients who have white lungs who will actually develop persistent fibrosis. Depending on the extent of the disease, this could be a massive fibrosis. It could also be that only certain regions of the lung become fibrotic and cause like a localized bronchiectasis, which is not really lung fibrosis per se, but can cause long-term problems for health, like chronic bacterial secondary infections can occur in regions of bronchiectasis. But again, this is too early to say. We have anecdotal cases of people we saw in the UZ hospital, where we saw people already with bronchiectasis 28 days after their first symptoms. So it can happen really fast, but we have to wait a couple of months before we know for sure. There's a lot of things on social media and on the internet where there are predictions, that this is going to be a massive wave of lung fibrosis in the future. I'm not so convinced of that, actually. It's going to be subtypes of patients.
Okay, then I would like to shift to Dr. Jamila Lawet. There were two questions for her. The first one is, how important is the exact formulation of the antigen with the adjuvant? And is that something that will be left to the antigen and final vaccine manufacturer to characterize and control for consistent interaction?
Yeah, so as any type of vaccines, we quite often make dose range escalations. The amount of adjuvant will be fixed because this is an adjuvant that has been used already in pandemia. The dose of the antigens will be tested, and we will pick the one that give the best immune response and the seroconversions. And this is going to be obviously a joint decisions of the two manufacturers because that would be a co-development. And I see another questions on the preliminary data in animal. The answer is yes. The data generated in model has shown clearly that combination seen by Clover of the adjuvants and their antigens and use a strong immune response, neutralizing antibody, which is way above natural infections, which is promising. And we hope that this will be translated also in clinical studies.
Okay, and then the final question is for you, do you have preliminary data and an animal challenge model for your vaccine?
So answer is yes, which demonstrate the induction of T cell and neutralizing antibody.
Since we have 3 minutes left, I would like to go back to Wim Ties because we have received one more question for Wim. The question is, how can you measure immunogenicity of your vaccine? If your immune response is local, will there be a systemic response as well?
We will look for systemic response, but more as a more general marker. Most of the analysis in the phase 1 trial will be done on nasal swabs. So we're actually also collaborating with the Leiden University, where there's a group who has been specializing in local immunity in the nose and using very not acceptable invasive nasal swab techniques where they can recover enough cells to do the analysis. And so we think that that system would be acceptable in the human volunteer trial, even repeatedly administered during the trial. So that's definitely going to be one of the key endpoints of the phase 1 trial.
Okay, and there was also a question if you could provide information on the mRNA doses that was used and the size of the mRNA carrier.
Yeah, for the moment in the mice, we're between 2 and 5 microgram in the nose for the mice. How it's going to translate in the human dose, that's still to be seen, but it's going to be definitely a bit more. Size of the carrier typically is 100 to 200 nanometer, but it's less important, we believe, especially for the spreading from the nose potentially to the lung. We believe that the particle size of the nebulizer is going to be more determined to see where actually the vaccine will end up, and not necessarily the particle size of the delivery system as such.
Okay, so by this we would like to conclude the Q&A session, and I would like to thank all the speakers and all the listeners, participants for all the questions that they have posed through the Q&A panel. For the last minute, I would like to give the word back to Sven Arnouds, Chairman of the Board, to give the concluding remarks. Sven, the floor is yours.
Yes, thank you, Veerle. And so this brings us to the end of this webinar session of two different webinars that we have organized. So first of all, I think it was a good experience, a first experience for us to organize these type of interactions with our network and with our members. and where we could show what these members are doing, the fantastic work that they are doing, and the insights that they have they have found at a rather short term. I would first thank also Fran and the Collegiate team who at a rather short term could organize these webinars. And then, of course, the speakers for the excellent work. So I want to thank them and congratulate them for the excellent work they did, also in a very short term, because we are only briefly confronted with this specific virus. And so for the very interesting presentations that they gave to us during these two webinars. And of course, I also want to thank all the participants for your contribution, for the questions you asked, which made this webinars also kind of interactive event. Now, maybe a downside of these webinars is that within a few moments, when we will push on the button and leave the meeting, that we are back alone at our desks, where usually we meet each other, we drink a glass and interact with participants and exchange ideas. Which for us is very important to get some feedback on what this meant for you, what were the good points, what were the maybe minor points, and which usually also provides us with new ideas to organize next events in the coming year. So please give us some feedback if you want, give us some ideas if you want, and use the mail address that is shown here, so info@flandersvaccine.be. And so with this, I thank you again for your participation, and I hope you can enjoy the rest of the day, enjoy your evening, and hope to see you on one of our next events. Thank you very much.